There is a big collection of Termitomyces heimii mushroom specimens in this trace. How long you took this to collect? Ms. Rosie D'Souza, my MSc senior research student, is here. This is my colleague of Department of Botany, Goa University at Taligaon, Goa. Campus which is known for its biodiversity, but which is also getting swamped by urban development all around us. So Termitomyces is a mutualistic mushroom with 72 recognized species in the world, out of which 35 species are found in India. They are all paleotropical species, and during my PhD in '93 I have done a lot of work on cataloging of this Termitomyces mushroom diversity, and we have been tracking also along with our ex-students who had done several projects on the mushrooms, the effect of climate change on fruiting periodicity or phenology of these species. And it has been increasingly observed that this species is fruiting now in the month of September, especially in Tiswadi Taluka in North Goa, because we have not reached other places. But it is quite apparent now that this mushroom, it is responding to climate change in a manner which is quite unpredictable. It has no independent existence, as you know. This is food of termite. This is the Theriomorph. Means this is the species. This is the Hymenium which discharges the basilospores. So this we call as lamine. So we have this type, we have the superior annulus, and we have this pseudorhiza. The pseudorhiza is not well developed in this, but we have got quite some good specimens. I think it is very difficult not to uproot the pseudorhiza because it is attached to the fungus gum. It is attached to the fungus gum. So you must have got quite a difficulty in removing some of these pseudorhizas and all. Here you can see that it is well exposed. This is a basal disc. Now what are the features? The identification of this species is Termitomyces heimii. Now this species was named by late Professor Natarajan, who was like a friend and guide to the mycological community and to me also, because he has encouraged me to work deeply in this area, taxonomy, ecology and biology and biodiversity of genus Termitomyces. And he published this paper in '75, and Roger Heim is the main person who has worked on genus Termitomyces and published a book in French in 1977, and he has been working in Africa and partly in Asia for more than 40 years. Roger Heim has worked on Termitomyces genus, and most of the knowledge that we have today in the world has come from like Dr. Roger Heim. So we owe everything to him. But what is remarkable about this species is that this has mutualistic existence with the mound building fungus cultivator termites, and that is what you observed. So you couldn't locate the fungus gum, it was difficult to locate. So how many clusters were formed means this must be having around 150 to 200 specimens, and there is a lot of variation. How many species you how many clusters you found in a orchard?
Around 10, sir.
Yeah, and how we define a cluster as such?
Cluster is like, it's a group. There is a group of mushroom which comes together. Like it is 3 to 4 more than that also. And it forms a super cluster. Okay.
Per cluster is a bigger patch of mushrooms.
Yeah, bigger patch. And
with different elevations. Some are short, some are tall, some are thick, some are thin.
Yes, sir.
Understood. But they look beautiful when you look at them. So must be done a lot of
field
work.
synchronous growth. It was not
Synchronous.
Opened.
You could do some documentation.
Yes,
Means basically how do we study Termitomyces in field? That is very important. That's what I accompanied her and I started teaching her there only. Means I said first we must have a map of that area because many clusters were coming up. This she divided into super cluster, clusters and mini cluster. A micro cluster could be having just one solitary specimen. Now it gives you an idea of the population biology of Termitomyces species, which is rarely studied. Now in lab you can study not only biology but also various morphometric species like we have here. As you might have seen that this is Termitomyces heimii. Let us take that this is the standard species. Now what do you call this? It is such a small species. You hold this, now you see the size variation. Now what causes this type of size variation? This is for us to explore in this department, in this lab, and this is the work which we have been doing. So my suggestion is that you do a lot of morphometric study. You take the measurements of this, you study the ornamentation, you see the pilial length, you see the pilial diameter, you see the pilial, the hormonal color, the thickness also. Then this is the stipe. Stipe is the epigeal portion. It is thick, solid, cylindrical and fibrous, and it subradicates. It tapers into a sort of pseudorhiza, because you see here, this is a beautiful specimen. Beautiful means the most ideal specimen for taxonomic studies. So what we see here is, you can see this is known as perforatorium or driller. This is a drilling organ. What is its role? Why it is black?
Because it passes through the thick layer of soil.
What is the chemical nature of that black pigment?
Melanin.
Correct. So the melanin is the same, only this is a fungal melanin. Melanin
Which is found
is present in this driller because it is not amenable to microbial attack. So this the whole epigeal part is a crucial stage in fruiting of this species, because you see this black part, it drills through the hard soil, comes up, and only then this hymenium develops. You can also see what is known as a collarium here. Collarium is that smooth, that circular part to which the lamella are free here. They are not attached to the stipe. Okay, they are free. So they are attached to a collarium. Collarium is a ring-like zone here. And there is a ring here, which is known as superior annulus. This is the remnant of this one. This is a universal vein. Understood? So this is basically the study of morphology, which is useful for taxonomic identification. Now slowly, as these lamellae mature, they turn pale pinkish. So my suggestion is that you have now got an excellent bumper harvest. Ecologically, it is very important, because she knows that these species do not appear during September. Today is September 28th, and we have never encountered the history of Goa. Termitomyces heimii, I am on record here. For 25 years I have studied this species. I am on record here that we have never recorded Termitomyces heimii species in Goa on September 28th. Now this is the first collection. So we must celebrate, because you know that you have got so much to study now. For example, you can do complete laboratory documentation by photographing this. Just now we saw size variation. This is one of the smallest and this is one of the largest. Are they identical species? That we have to investigate. Or they are forms and varieties of Termitomyces heimii? Very little knowledge is there. Because on Index Fungorum or in mycological index, we will find lots of forms and varieties of genus Termitomyces also. But truly recognized species may be actually less, because it has a very interesting life cycle. You see this part, this is the underground part. So what is the role of this organ? What it is known? Yeah, pseudo means false. So it is a root-like organ, it is not true. So it is a basically nutrient translocating organ which develops from a basal disc. Now this basal disc is attached to a fungus core. Now what is the composition of the fungus core?
Fungus core, it is the termites are they get their saliva and the soil particles, and they grind it, and it extracts and out and it forms that fungus core.
What is the main component of fungus core, which is mineralized? Which is from where they get the main component?
The leader.
Yeah, from where they transport this to the fungus core. Basically, it is dead organic matter which the foraging termites collect and transport through various tunnels which connect to the fungus comb galleries, and then they pile it up in a brain-shaped, cerebroid-shaped mass, and then they tend the fungus garden. Now that stage genetically is known as Termitomyces sporodocial anomorph. So the anomorph doesn't fruit. So only when conditions are ideal. I don't know on September 20th what are ideal conditions, because this development takes minimum 8 to 10 days from this to this stage. Then another 4 days for this. So 15 days it takes for full growth. That is for holomorph. So we have the underground anomorph, sporodocial, and we have this teleomorph. Now what is the role of teleomorph?
Teleomorph to release the
basidiospores. So basidiospores fall, and then the termites collect them and then inoculate on the fungus garden. So you have got some ecological idea. Let us now see what are the potentialities of biological and morphological studies. For example, there is very little knowledge about the population studies, means what is the variation. So you can do morphometry, that is take various measurements, also weigh the specimens individually and take biomass, understood. And you can do studies on the driller also, you see this. We have very little knowledge on the driller. Then we need to see the variation statistically, and besides, we had hardly any information on chemical, biochemical composition of this pileal tissue, hymeneal tissue, style tissue and pseudoresal tissue. So I expect you to sacrifice all this, chop this, collect pseudoresal, chop here, collect all style, remove this lamellar part, understood, and then you can grind it and prepare extracts, so that these extracts could be further subjected to chemical, biochemical analysis to detect certain minerals, sodium, potassium, calcium, iron, manganese, etc. Understood. So you can proceed further, and also simultaneously you can take some excellent spore prints, because then you told me that there are some insects associated with some of these specimens. What are these?
There were beetles, snails, snails also.
What is possibly the role of beetles in these mushrooms, according to you?
They might
play their eggs, feeding here. So their life cycle may be associated.
Yeah, associated.
Besides the insects,
Eggs also I found, sir.
Yeah, and then?
And snails.
Snails. So snails are known, but what snails actually damage the tissue. I don't know whether the snails are associated, but you can see now lots of insects are there within this. You can see. Yeah, you can see this beautiful guy here. Some people will find it ugly, but I find that bug is now, it has come there. It may be a female. It is trying to lay the eggs there within the tissue, because the hatchlings will be, that decomposed tissue will be consumed by the hatchlings. Because if there are rains, these mushrooms would have decomposed. It is good that I alerted you and went there and made a good collection. So there is entire life cycle of, in the food chain which is associated with this fungal biomass. Because this is nutrient rich. These are edible species of Goa. So if you have to left behind this entire collection, people would have just come and collected, and they would have consumed. What type of preparations you make in Goa from these mushrooms? Because I think at these stages people are consuming.
Yeah.
So what do you make it? Do you eat this? Do you consume this?
Yes, sir.
So at what this is the stage which comes in market, isn't it?
Yeah.
What
Destroy that we get.
That is there, but is it delicious?
Yeah, it is.
So what preparations could be made from this? Means because see, now we have to also discuss food security of the world. The largest species in this is Termitomyces titanicus, which weighs up to 2 kilograms, and its diameter, now you see that this is just about some 12 centimeters, 10 to 12 centimeters, and this is heimii. But if you go to Zambia, you will get Termitomyces titanicus is a diameter of 80 centimeter to 1 meter, and you can carry it like an umbrella. Now I can't carry it like an umbrella because it is so small, but perhaps you can carry it like an umbrella. You see. There are photographs in Zambia, Termitomyces titanicus, and then you can bring that umbrella at home. It is probably world's largest edible umbrella. You can cook it and eat it. So world is looking for alternative sources of food, because good fertile agricultural lands are running out. And it is possible through genetic engineering techniques and all. Suppose the genes which code for large, species, if you use those genes, understood, then it is possible to grow larger and giant mushrooms. And if we can perhaps use these Termitomyces genes, which are very useful, because this is Termitomyces also as established by Indian Institute of Chemical Biology, Kolkata. They have done a lot of research since last 40 years, and they have found that there are many types of hydrolytic enzymes which are produced by these species. So these Chinese scientists have also found brain activating chemicals, neurotogenic cerebrosides. So this is actually a source of pharmaceuticals, new age medicines, edible proteins, because I think you can even cultivate this in a liquid culture, and you can have mycoprotein also, Termitomyces, texturized. So all these possibilities are there. That's why we need to take these genus and the species which people are just picking from the wild and destroying the diversity very seriously. So I leave it to my student Miss Rosie D'Souza to carry the torch forward, and I thank you, Miss Priyanka Shirodkar, for sparing your time for this documentation, because I believe that in our campus itself This is a gift of God, I would say, this gift of Mother Nature, that suddenly on 20th September she decided to help us in our project. She is working on Termitomyces project, but she was telling what you were saying, you were not hopeful of seeing a good enough rectification.
Yes.
Now right in front of your door, the Mother Nature has produced now a bountiful of this. And the Mother Nature is perhaps appealing to you that please do justice to me, I have given you so much. Now it is up to you to use the knowledge which is there in front of you. So with this message, I think we will also tell the world, because this video clip is specifically taken for uploading on YouTube, just as previous YouTube video clips, with the hope and message that we cannot take global biodiversity for granted. Climate change is causing some favorable changes also in fruiting periodicity of macromycetes like tropical macromycetes, mutualistic basidiomycetes like Termitomyces, and this is a big challenge also in terms of creating food security, because we can very intelligently use climate change, and other factors to discover what is it which triggers this unusual fruiting. And if we crack that secret, it is possible that artificially these termite mycoproteins can be produced, and since they are rich in macro and micronutrients and proteins, I think the world can look forward to this genus as a potential passport for food building, food security based on fungal mycoproteins. I think for everyone in this world there is something in this genus. That's why this is worshipped in Goa. Understood? There is a goddess of termite hill which is known as Santeri. Understood? So let us consider this as a gift of goddess Santeri, and let us enjoy the knowledge, and I think you have a lot of work at hand. So let us thank Mother Nature and thank Ms. Priyanka Shirodkar also for being patient to take this documentary. Thank you very much.